Students, welcome back to our biology class. Today we can discuss the structure of DNA. Before learning the structure of DNA, just we can discuss the history behind the DNA. Do you know who discovered DNA? Discovery of the DNA double helix. First one, Frederick Griffith, 1928, discovered that a factor in diseased bacteria can transform harmless bacteria into deadly bacteria. That means one factor is making a harmless bacteria into deadly bacteria. That factor we can call as DNA. This is discovered by Frederick Griffith. Later on, Morris Wilkins worked on the structure of DNA. Thereafter, Rosalind Franklin 1952 worked out helical structure of DNA by X-ray photo of DNA. Finally, Watson and Crick described the double helix structure of DNA, for which they were awarded the Nobel Prize in 1962. A single DNA molecule is very large and hence it is described as a macromolecules. It is composed of two complementary strands wound around each other in a double helix structure. Each single strand is composed of repeating unit. That repeating unit of DNA strand we can call it as nucleotide. This nucleotide itself having three components. What are they? First one is a phosphate group. Second one is sugar unit, that is pentose sugar, and third one is a nitrogenous base. These are the three components of a single DNA strand. So this is the phosphate group and this is the sugar unit and these are nitrogenous bases. By these three components only nucleotides made of. Next we can discuss what are the four nitrogenous bases present in this DNA. There are four nitrogenous bases. First one is adenine base, second one is guanine base, and third one is thymine base, and fourth one is cytosine base. These are the four nitrogenous bases present in the DNA strands. These bases are connected by a hydrogen bond. These bases are connected by a hydrogen bond. Then adenine connected with the come to the point. Adenine connected with the thymine with a double hydrogen bond, whereas guanine connected with the cytosine with a triple hydrogen bond. Once again, adenine connected with the thymine with a double hydrogen bond, whereas guanine connected with the cytosine with a triple hydrogen bonds. Okay, these are the bonding of these nitrogen bases. Next we can discuss structure of nucleosome. What is a nucleosome? The DNA strand winds around a core of 8 histone protein. Each such a complex is called a nucleosome. This is the histone protein. How many histone proteins are there? 8 histone proteins are there in this. Like this way, each DNA strands wind around a core of 8 histone protein and make a nucleosome. An individual nucleosome can be imagined like a football around which a long rope is wound with 1 or 2 loops. Let us revise the topic once again. Some question I can ask. First one is, who proposed the double helix structure of DNA? Watson and Crick proposed double helix structure of DNA. Second question, what is a nucleotide? Each single DNA strand is composed of repeating unit. That repeating unit we can call as nucleotides. Next question, what are the 3 components of nucleotide? The 3 components of nucleotides are phosphate, pentose sugar and nitrogenous base. Next question, what are the 4 nitrogenous bases present in the DNA strand? The 4 nitrogenous bases present in the DNA strands are adenine, guanine, thymine and cytosine. Adenine connected with thymine with a double hydrogen bond, whereas guanine connected with cytosine with a triple hydrogen bond. Next question, what is a nucleosome? The DNA strand wind around a core of 8 histone protein. Each such a complex is called a nucleosome. Is it clear? So these are the some important questions can come from this topic. Learn this very clearly and I will come with the next topic in next class or in next video. Till then, bye.